Today I'm presenting on capture myopathy, a deadly condition that is very relevant to those of us whose work requires us to handle or observe wild and non-domesticated animals. Whether you're on the rescue side and frequently handle and care for wild animals, or in large-scale conservation with less direct or frequent contact, we all need to be acutely aware of how the stress of our interaction can negatively impact a wild animal. This cute bunny makes a great point. Bunnies are very sensitive to stress and known to die from handling, but we see this condition in many animals, terrestrial and aquatic mammals, birds and reptiles, and in ungulates and prey species especially. I've noted translocation. Much of the available literature on capture myopathy is from conservation efforts to move populations. These programs are vital to species survival and ecology, but struggle with animal deaths during capture, transport, release, or sometime thereafter. Capture myopathy causes severe pain and loss of bodily control. Animals can suffer mere minutes or weeks before death. Treatment is limited to supportive care and the hope that the animal regains homeostasis. And even then, their body condition or ability to mount future stress responses may not recover, so prevention is key. And to do that, we need to address how it occurs. Acute stress is a beneficial response that helps animals deal with threats. We've all heard of fight or flight, which starts in the brain with a distress signal from the amygdala to the hypothalamus and activation of the sympathetic nervous system. Nerve signals using norepinephrine stimulate the adrenal medulla to release epinephrine into the bloodstream. This increases heart rate, improves airflow to the lungs, encourages fuel stores to convert to readily available glucose, and diverts blood from non-essential systems to the skeletal muscles. After the surge of catecholamines, the hypothalamic pituitary axis, or HPA, the endocrine component, kicks in. The hypothalamus releases corticotropin releasing factor into the portal system shared with the anterior pituitary, which secretes adrenocorticotropic hormone, ACTH. ACTH stimulates the adrenal cortex release of glucocorticoids, which prolong the conversion and availability of glucose for muscle metabolism and maintain blood pressure. These are all good things when you're immediately trying not to die. Chronic stress, though, either prolonged threat perception or a period of multiple acute stressors, is detrimental. Continuous elevation in catecholamine levels can cause cardiovascular damage, and sympathetic overstimulation keeps muscles tensed. Normally, when glucocorticoid levels get too high, this signals the HPA to stop stimulating the adrenal glands. But when the HPA is overworked, what researchers have seen is the output of cortisol or corticosterone lowers just a bit, so the upstream signals do not stop, and sustained glucocorticoid levels start to damage systems. The nervous system is overstimulated, and muscles become exhausted. Rhabdomyolysis is the muscle damage that characterizes capture myopathy. This is from the exertion of evading pursuit, capture, or handling, or from injury inflicted during those events. And in some very large mammals, it occurs when they are immobilized in a recumbent position. At the cellular level, first, prolonged nervous and endocrine responses heighten muscle contraction, which uses energy in the form of ATP. As ATP is depleted, energy-dependent ion pumps that regulate calcium levels inside the muscle cells start to fail. Increased calcium activates phospholipase and protease enzymes that break down cellular membranes, mitochondrial membranes included. And with fewer mitochondria, we see a shift towards glycolytic metabolism, which accumulates lactate. This buildup is further aggravated by adrenergic stimulation of glycogen breakdown into even more glucose for glycolysis. Next, degradation of the muscle fiber membranes allows fluids and sodium from the blood into the cells. Sodium influx heightens muscle contractions by triggering calcium release from the sarcoplasmic reticulum. Removal of water from the blood also causes hypovolemia. Eventually, cells begin to lyse or burst, at which point their contents flood the bloodstream. These are built up lactic acid, which decreases blood pH, myoglobin, creatine kinase, potassium, and phosphate. Importantly, high levels of potassium can disrupt cardiac conduction and cause irregular heartbeats and even cardiac arrest. Hyperthermia is also common in capture myopathy. The exact mechanism of excessive endothermy is not well understood, but Powers et al. Has proposed that the buildup of reactive oxygen species from disrupted oxidative phosphorylation results in heat production and cell death. Muscle damage byproducts lead to myoglobinuria. In small amounts, myoglobin is carried to the liver by globin proteins. Once these are saturated, the kidneys step in to clear the rest, but they are overworked by high quantities. Myoglobin also reacts with proteins in the kidney nephron tubules to form brown granular casts. These plug up the tubules, lowering the volume of liquid filtered into the bladder and creating high-pressure regions upstream of the obstructions. I had mentioned hypovolemia from fluid movement into the muscles. The central nervous system responds to low blood pressure with vasopressin, a hormone that constricts blood vessels. This limits blood flow to the kidney, elevates its internal pressure, and further depresses filtration. Animals with myoglobinuria end up with swollen, dark red kidneys and bladders that are empty or contain only a small amount of red-brown urine. Continued obstruction of the tubules leads to progressive necrosis and death from acute kidney injury. A useful mindset in our animal interactions is to treat stress as a predictable factor and aim to reduce the number or magnitude of perceived acute stress events. Engage with the fewest number of animals using the minimum species trained staff for the success of the job at hand. Always limit noise, smells, and visual cues such as light and movement. Chemical restraint is great if the stress of sedation would be perceived as less stressful than events experienced while conscious. Avoid drugs like opioids that stimulate the sympathetic nervous system. For transport, consider vehicle size and climate control, particularly for our ectotherms. Ensure high-quality driving and minimize travel duration. Captive settings should imitate the natural environment, and human visits are kept to a minimum or on a predictable schedule to reduce stress intensity. Non-invasive health checks, like fecal hormone measurements, can indicate average stress levels. Fluid therapy may be able to alleviate hyperthermia and hypotension. For release sites, take into account food availability, predation, and population density of conspecifics, and release at the least stressful time, during an easy season of the year and during the time of the day the animal is typically active. Lastly, trail cameras and satellite monitoring are great research tools that decrease repeated interaction. These techniques are constrained by cost and logistics, but advanced planning where possible can help mitigate some of that burden. And at the end of the day, animal welfare is our goal and duty.